So welcome everybody to our webinar today. We are really happy that you join our invitation and that we can listen to two additional talks today. After the talks yesterday about the cultivation of bacteria, methanogens, and protozoa from the rumen, we will continue today with fungi and the phages and the viruses. And we have two excellent speakers for this for you. And I would like to first introduce you the first speaker, which is Yanfeng Cheng. She is a professor at the Nanjing Agricultural University in China. And during her PhD studies, she visited Aberystwyth and worked with Mike Theodora and John Edwards on fungi and on the co-culture with methanogens. Now, back in China, she continued the work using or characterizing co-cultures of anaerobic fungi and methanogens, and she was able to isolate and characterize more than 30 anaerobic fungus methanogen co-cultures from the rumen and from feces of different herbivores. The main research actually now is to study the interaction of the anaerobic fungi and the methanogens for the process of fiber degradation and methane production, with a specific focus on identifying robust cellulose and hemicellulose degrading enzyme. So Yanfeng, we are looking forward to your talk. Please, can you open the presentation mode in the PowerPoint? Because we still see the presenters mode. Yes, now it's perfect.
Yeah, okay. Yeah.
And I ask everybody to type in the questions in the Q&A box. Thank you very much.
Okay, thank you, Yana, for your introduction. And good morning, everyone, and good afternoon in China. And I'm Yanfeng Chen at Nanjing Agricultural University. Today, I would like to introduce some of my experience on anaerobic fungi and their role in the rumen. So we know that in the rumen, the most important function is to degrade the plant materials that the animals are ingested. So during the process of plant degradation, bacteria, protozoa, anaerobic fungi, they degrade these plant materials to volatile fat acids. For anaerobic fungi, they are famous for their ability to convert large particles to small ones. And then at the meantime, they produce large amounts of hydrogen. This hydrogen can be then utilized by methanogens to produce methane. So in the rumen, anaerobic fungi and the bacteria and protozoa, they are very important for the degradation of plant materials. And what is anaerobic fungi, and what's the difference between anaerobic fungi and aerobic fungi? And in the domain of fungi, anaerobic fungi, they are an independent phylum, and they are very, very different from aerobic fungi. So the first one is the genome size. We can say that the aerobic fungi, the genome size is something like 40 megabase. And for the anaerobic fungi, the genome size is much larger. And so far, we have 12 anaerobic fungal genome. And the minimum size is a bit more than 70 megabases, and the largest one is about 200. And the second one is the GC content. So we know that the normal GC content is around 50%. Okay, for aerobic fungi, the GC content is around 50. However, for anaerobic fungi, the average GC content of the genome is about 16 to 17. And what's it mean? There are some fragments, there are some proportions in the genome of anaerobic fungi. Their percentage is about 50. And that means there are some proportions of the genome, they have really, really low GC content. According to our experience in genome sequencing, the lowest GC content in the fungal genome is less than 10. So in some regions, the GC content is about 8 or 9%. And the last one is the protein coding regions. It's the anaerobic fungal protein coding regions are much lower than aerobic fungi. So there are many researchers on the world. So the first one is Orpin. If you are interested in anaerobic fungi, you might know him. And the next, and all over the world, there are many, many fungal researchers. They are from many from UK, and some from New Zealand, from US and India and Austria, German, Czech, and also Canada, and of course China. So far, there are 22 genera identified. We can say from here that before 2001, there were only 6 genera identified. However, over the last 10 years, there are 16 genera identified. So of course, over the last 10 years, there are much, much more researchers interested in anaerobic fungi because of their ability, high ability in lignal plant materials, in the degradation of plant materials. So that's the main introduction of anaerobic fungi. And we know that over the last 10 years, with the development of high throughput sequencing techniques, and there are many, many papers or data with those sequencing. And we have the many, many metagenomic or metatranscriptomic data on rumen samples or phase samples. However, we can say that there are very, very low percentage of fungal genome in this data. And in some cases, there are even no fungal genes in those data. And the other thing is the marker gene analysis, because we know that ITS or LUS or IGS sequencing, with those marker gene analysis, we can know some fungal community in the samples. However, the OTUs or ASVs are relatively lower than what we expected. And in some cases, we can not amplify the fungal genes. So, for the cultivation of anaerobic fungi, it's very important for us to look into the function of these microbes in the rumen. And so I will introduce the cultivation of anaerobic fungi in these 4 parts. The first one is enrichment and isolation, purification, characterization, and also genome sequencing. So the first one is enrichment. So if we want to get the pure culture for anaerobic fungi, we need to get the enrichment first. And the first thing, the very, very important thing is the media. So we need to prepare the media in a correct way. Otherwise, the fungi could not grow. Okay, so it's very important. And the second one is the samples. So we need to collect the samples and then inoculate, dilute, and inoculate into the media as soon as possible. Because because if we expose the samples in the air for 2 hours, 4 hours, and then the fungal might not grow in the culture. And after 3 days, maximum 5 days, there are some of, you can say that the floating of plant materials, that means the fungi grow in the culture. So the most important thing for the cultivation, for the enrichment of fungi is that we need to inhibit the growth of bacteria, because we know that there are large numbers of bacteria in the samples, rumen or feces. And if we cannot inhibit the growth of bacteria, the fungi would not grow. Okay, so sometimes we can use chloramphenicol to inhibit both bacteria and methogens, and sometimes we use the streptomycin to inhibit only bacteria. So it's up to you for your purpose of the isolation. And after you get the enrichment, it means that you have a diverse of anaerobic fungi in the enrichment. And then the next step is to purify to get the monoculture of the fungi. And then usually we use the rolling tube technique, because yesterday there was some so there is introduction on rolling tube, so I would not introduce very in detail. We use the rolling tube to get the colony and then pick them into the liquid media. So then after several times of pure rolling tube of the purification, and then we will get a pure culture. So after we get the pure culture, we need to categorize what fungi are they. And there are 2 publications so the first one is a review. The authors, they compared, they described all the current texts in this review, and then based on this characterization and this description, the authors from NOHA's group and also some anaerobic fungal network members, we propose that a new assignment criteria if you want to assign a new genre. So if you want to know more details about the characterization, you can have a look at these 2 papers. So briefly, there are 2 ways to characterize what your fungal strains. The first one is the morphology and the growth. So according to the morphology, you can say the colony on the rod tube. So they are different size or different phase. And also the juice balls. There are many especially the fragments of the number of fragments of the juice balls, and also the rhizoids, and also the sporangia, the size, and also the phase of the sporangia, and also the substrates and the end products. So according to this morphology and growth characterization, and then with the phylogenetic analysis of the fungal genes, microgenes, and then we can identify what genre they are belonging to. For the phylogenetic analysis, we usually use both ITS1 and D1 and D2 regions. So sometimes if you cannot identify them with these 2 sequences, and then we can use the whole length of ITS1 to D1 and D2, because we know that the genome, the similarity of the genome sequences are is very, very high. So it might be better if we can use the whole sequence of from ITS1 to D1 and D2. And next one is the genome sequencing. And after we get the pure culture, we need to store it, okay? So the storage of a number of fungi is now actually a problem. And usually we need to store them at -80 for short-term storage and liquid nitrogen for long-term storage. So please remember that do not store at -20 because you won't get a live fungi if you store them at -20. And after that, you need to think about deposit your strain, your culture in a collection. And now there is several collection. One is in German, and they are famous, okay, DSMC. And the second one is HCC. And in China, we also have a collection in Beijing. And in our group, we usually deposit our anaerobic fungal strains in this collection. And however, so far there is some problem that we need to fix is the characterization of our strains in these collections. Because according to our experience, we deposit several genera in this collection, and then they identified the strains as neocharismatics. And then for genome sequencing, we need to pay attention. The first one is sample collection. So if we want to do genome sequencing, we cannot use the open media, which include rumen fluid. And we need to use media B, and also fresh samples are recommended. And if you cannot extract DNA, after you collected samples, you need to store them at -80 or liquid nitrogen. And next one is the DNA extraction. Remember to use agents to prevent the degradation of DNA, because fungal DNAs are really, really easy to be degraded. And don't store them at -20, because if you store them at -20, and you will lose your DNA, okay? And so we recommend that you need to use your fresh DNA for your sequencing. Do not store them at -20, even -80 or liquid nitrogen. For the sequencing, because we know the low GC content of the genome sequencing, and we recommend that we need to shorten the time period from DNA extraction to library construction and to sequencing. That means once you get your samples, you need to extract the DNA. And once you get with DNAs, you need to sequence your data, your samples. So, because there are really, really high aid content in the genome. So after that, I will introduce the role of the fungi in the rumen. We know that in the rumen, the most important thing is to degrade the plant material. So the largest amount of fiber in the plant material. So the fungal has very high diverse of enzymes that degrade the plant fiber. And the second one is some anti-nutritional factors in the plant fiber, because we know over the process of growth, the plant need to resist those pesticides. So they will excrete some some chemicals that resist the pests. So these chemicals, they are anti-nutritional factors for animals, and they can prevent the degradation of microbes in the rumen. So some of these microbes in the rumen, they can degrade those chemicals. For example, the tannin in the plant fiber. So that's in our opinion, that's the second important role of fungi in the rumen. And the notes for fungal isolation, the fungal has the the fungal has very sensitive to is very sensitive to oxygen. So during the whole process of our operation, we need to reduce the handling time as much as we can. And the second one is contamination, especially the bacteria. We need to keep in mind that to inhibit as much bacteria as we can. And the second one is the temperature. The anabolic fungi is very sensitive to temperature. So be sure that to keep the fungi at 39 centigrade. And according to our experience, the co-cultured methogens would increase the fungal resistance to stress, for example, to oxygen, to temperature. So in our lab, we usually use penicillin and streptomycin to inhibit only bacteria and to get the enrichment of fungi and methogens. In this case, the fungi might grow much better than we use chloramphenicol. Because if we use chloramphenicol in the enrichment, we will inhibit both bacteria and the methogens. So if you want to get more information on anabolic fungi, you can visit the anabolic fungi network website, and also we have Twitter, Facebook, Instagram, and WeChat platform. And acknowledgement, thank my supervisor and my students, and also the funding. And questions are welcome.
Thank you very much, Yanfen. So please type your questions into the QA box. There's already 2 questions. Do you see them, Yanfen?
I can. Yes. So the first one is to preserve room samples for days before transfer into laboratory for cultivation. Actually, we do not preserve room samples for days. So if we want to cultivate anabolic fungi, we usually take our bottles, take our media to the farm, and then we need to dilute and inoculate the samples, the samples into the medium immediately after a sample collection, and then keep them at 39 centigrade, and then transport to the laboratory. So we did dilution and inoculation just on site. And the second one is, do you need to put the pure culture in polar solvent before storing at -80? No, we don't. We add DSMO into the fresh media and then put them into -80 immediately. After overnight of -80, and then we transfer to liquid nitrogen. We do not. We use the DSMO for the storage. And next one is, what is the relationship between fungi and the methogens? And according to our experience, this relationship, they benefit for both anaerobic fungi and methogens. So for anaerobic fungi, they can survive in low temperature, for example, at 4 centigrade. So if you have the co-culture of anaerobic fungi and methogens, if you store them in the fridge at 4 centigrade, and then they can survive overnight. But if there is no methogens, then they would die. So the methogens, they help the fungi to resist the stress. And the second way is the methogens, they improve the fiber degrading ability with a very higher, let me say more than 10% of fiber degradation ability. And for the methallogens, if you culture the anaerobic fungi with methallogens, and the methallogens would grow very well within 3 days. That means the anaerobic fungi, they might provide some like grown like factors for those methallogens. And so what's those growth factors? Actually, I don't know. We tried to analyze the supernatant of the culture, but until now we do not know what's the growth factors. And also there are much more methallogens, so more diverse of methallogens in the co-culture, comparing with those methallogens pure culture. So I want to transfer samples from Nigeria to the UK for cultivation and food analysis. I think it is not possible. I don't think it's possible for you to transfer your samples from Nigeria to UK, because so there is one way. If you get the phases. And dry the phases in a very quick way, and then the fungi, they might be preserved. And then you take the dried phases from Nigeria to UK, you might get the cultivation of fungi, because according to some researchers in UK, they found that there are some rhizoids or aspergillus, they can survive in the phases. But the most important thing is that you need to dry them very quickly and to preserve them. Which is the best period of time to replicate the cultures to maintain the fungi alive? So for the cultivation in the lab, we usually transfer them 3 days. So for liquid nitrogen, so far as our experience, we have fungal strains more than 3 years we can survive from the liquid nitrogen. So if you store the store your fungal strains in liquid nitrogen, you don't need to transfer them within 3 years. But for your cultivation and 3 days, we usually transfer 3 days to maintain them. So what dilutions would you recommend for the isolation? Usually we dilute the rumen samples from -1 to -5, 10 to -5, and then inoculate -2, 3, 4, 5 into media. And then probably you can get the fungal the fungal culture from let me say -3 to -4. Sometimes you can get also fungal fungi in the -1, but in -1 there are many bacteria. So we usually choose like -3 or -4. Are the fungal culture producing hydrogen? Sure, of course, because the fungi, they have hydrogenosome, and then they produce large amounts of hydrogen, especially when they co-cultured with methallogens. The metabolism in the hydrogenosome, they were improved. It is improved dramatically to produce much more hydrogen to transfer to methallogens to produce methane. Can the supernatant of AF used to reach methallogens? It's a good question. I tried to use supernatant of anaerobic fungi to enrich methallogens. Of course, in our enrichment of the anaerobic fungi and methogens, we found a novel methogens, and then we tried to use the supernatant of anaerobic fungi to get the novel methogen culture, but we failed. So if you want to enrich methogens, there are many media for methogens. How long is the short term in general when samples were stored at -80? In our experience, we store our culture. So you mean the culture or rumen samples? So for the culture, we did use them a story at -80 for 1 month. And for rumen samples, for metagenomic analysis, it depends. If you do not analyze anaerobic fungi, and you can store them for a long time. But if you want to do anaerobic fungal molecule analysis, I would recommend that you do DNA RNA extraction as soon as possible you get the samples. At what temperature do I need to dry the sample before transfer to UK? No more than 40 centigrade, otherwise the fungi might die. Or is it possible to isolate the fungi and then transfer the culture in liquid nitrogen to UK for analysis? I'm not sure the rules between these two countries, because in China, so there is a group in German, they request fungal DNAs from China. I tried to ship the DNAs from China to German, but failed, because the rules, the shipping rules. So I'm not sure. You need to refer to the rules, and I would suggest that you do the analysis in Niger and then send the data to UK. Or can I put the samples in glycerol? Yes. At the most beginning, people use glycerol to store anaerobic fungi, but after that, we found that DMSO is much better, so we usually use DSMO with 5%. How would you recommend to store anaerobic fungal DNA if not used directly? It's a good question. If you do not use it directly, I would recommend that you store the fungal cells instead of store fungal DNAs.
Okay, cool. So there was a lot of questions. You see, it's a really hot topic, Yan-Fen, which you're working in. Just as a comment for this discussions about shipping of Nigeria, UK, you have to follow the Nagoya Protocol for transferring biological samples. It's really critical nowadays. We also observe this, so please also remind on this that we have this Nagoya Protocol, which is not so easy to transfer cultures or even rumen samples from one country to another one. So that's the thing we have to deal with at the moment. Cool. So there are no other questions. Yan-Fen, thank you very much for your excellent presentation. It gives us a lot of new information about how to handle the anaerobic fungi.
Thank you.
And we will continue with another hot topic, and it's about the viruses and the bacteriophages. And we are really happy to welcome Ross Gilbert from Australia. So good evening to you. Ross is a microbiologist which is hosted at the Queensland government in the Department of Agriculture and Fisheries, which is based at the Ecoscience Precinct in Brisbane. And Ross has a keen interest in using innovative science to better understand the role of the microbiome in ruminant livestock production systems, with a particular focus on improving productivity and reducing enteric methane emission. Russ obtained a PhD on factors affecting bacteriophages of the rumen ecosystem. So she's already dealing since a lot of, or 3 decades, I think, at least already, with bacteriophages in the rumen. And she has undertaken a postdoctoral position at the University of Glasgow in the Division of Virology. And she was at the Zero Livestock Industries. And Ross has been now in the Queensland government since 15 years and is a principal research scientist with the microbiology group. So Ross, we are really happy to have you here, and we are looking forward for your talk.
Okay. Screen. Share. Okay, can you see my presentation okay there?
We just see the presenter mode.
Oh,
Maybe you can change the display settings. Yeah. Yeah, now it's fine. Perfect.
Better?
Thank you.
Awesome.
Yes.
Thank you so much for that kind introduction. And thank you to the GRA organisers for inviting me to talk and present today. So I put up the short version of my title, but the long version is what was suggested to me in the first place. So today I'm going to talk about techniques to study viruses and bacteriophages, how old approaches have been improved, and what we still need to work on. So just by way of background, I work with the Microbial Ecology Group in the Department of Agriculture and Fisheries with the Queensland State Government. So the group has been running since the 1990s with a focus on rumen microbiology research. Australia has historically had a very strong track record in rumen microbiology research due to its importance to agriculture and ruminant livestock industries, the importance to the Australian economy. The state of Queensland has a relatively large land area and a range of climatic bioregions, encompassing a tropical northern region, warm humid coastal regions, and a usually dry interior, and a temperate southern region, which is where we're based. The most common beef cattle breeds in Queensland are Brahman or European crossbreeds, with the introduced Bos indicus traits providing insect resistance and better heat tolerance. So Australia's beef herd is around 22 million head, and Queensland has Australia's largest beef herd of approximately 10.8 million head. With around 90% of these raised in extensive grazing systems and some grain feedlots. Reducing enteric methane emissions from this ruminant livestock base is really important both to the red meat industry, and it's a government priority area as well. So our research area efforts are focused on ways to better deliver methane reducing strategies to the farming systems of Queensland and Northern Australia. So I understand that this webinar series has already covered the major microbial groups found in the rumen. So that's the prokaryotes, the bacteria and the archaea, and the eukaryotes. We've just heard a lovely talk about the fungi, but there's also protozoa there as well. So my special interest in the rumen really has always been in rumen phage. So initially, the viruses in the rumen were thought to be transient, possibly ingested through feed, but that idea didn't really last very long as the advent of electron microscopy as well as culture-based techniques showed that the rumen contained a large, diverse population of viruses, and they predated on and replicate in the microbial populations. So if you take a drop of rumen fluid, as I have here, and look at an electron microscope, you'll see a wide variety of virus types. So just a bit of terminology, like for many years, rumen viral populations were only really referred to as bacteriophages. Phage means to consume, and bacteriophage are viruses that consume bacteria. Similarly, viruses infecting archaea were called archaeophages. These days, viruses infecting archaea are usually simply referred to as viruses, as are the viruses infecting eukaryotes. It can get a little bit confusing at times, and researchers have their own way of saying things. However, the bacterial virus researchers seem to have stuck to their phage terminology, and it's what I tend to use as well. So people often start their research papers saying that there hasn't been very much done on rumen phage research. And compared to the efforts put into characterising rumen bacteria and fungi, say, that's absolutely true. But there has been a slow and steady build-up of knowledge in the phage space. And research efforts can really be chronologically linked to developments in technology that's available at the time. So this graph at the top includes 110 publications in books and journals relating to rumen phages and viral populations. The record spans almost 60 years, beginning with the first short report by Adams from the Iowa State University in the 1966. So electron microscopy enabled phages to be visualized for the first time, and several groups undertook morphological surveys and performed counts to see how many were there. These studies showed that most phages are tailed. They are now classified. There's been a very big change in viral classification even in the last couple of years. So now these tailed viruses are classified in the class Caudovirus. So the cauda is Latin for tail. At this time, people began to speculate on how phages might affect rumen function, particularly their role in the lysis of rumen bacteria. So molecular biology led to the use of pulse field gels to enumerate and profile rumen phage populations, and for the first time really showed breed and diet-based differences in the populations. DNA sequencing was in its infancy during this time, and it was time-consuming and used lots of nasty chemicals. But that didn't really hold back the rumen microbiologists. They sequenced rumen virus genes and used them in cloning vectors for the transformation of rumen bacteria. Following this, after the studies by Dinsdale, Wilner, and Berg-Miller in Brian White's lab, virome studies are really taken off, and they've dramatically increased our ability to look at phage genes. There's also been a nice proteomics study by Phil Pope's lab, but most of the reported culture-based phage isolation work was really undertaken in the 1960s, 70s, and 80s. And some of these phage collections might just be waiting for institutions to revisit them, or the collections, unfortunately, may no longer be in existence. So in the last 10 years, genome sequences have been obtained for a few phage particles infecting rumen bacteria. And apologies if I've missed anybody study here, but I don't think I have. So they include phages from a historical phage collection. Our group has been looking after this collection for many years, and some of these were sequenced as part of the Hungate 1000 project. And a couple of other culture-based studies have been published, which is great to see. Butyrovibrio is such a common rumen bacteria. It's nice to see these published phages, and the Park study included some phage biology as well. So this list of hosts brings me back to the culture-based element of this talk, because when it comes to culturing phages, you really need to know and understand the cultural requirements of your host. I'll just touch on a bit of phage biology, which some of you may already know, but I'll just go over it anyway. So there are a few different replication strategies that they can adopt. With phages, there's almost always exceptions to the rules, but to keep things simple, there are two main replication strategies, lytic and lysogenic. Both replication strategies start with infection. So that's in the middle of this diagram here. The attachment or absorption of a phage particle to a host cell. Phage DNA enters the cell and the lytic cycle can commence. So essentially the phage is taking over the host cell and turning it into a phage factory to produce components which then assemble into phage progeny particles. They're released by bursting or lysing the host cell. Alternatively, the phage infects the cell. Phage DNA either circularizes, like making a little plasmid or episome, or integrates into the host genome. The phage encodes a cassette of genes that enables this integration together with replication control genes, like a series of repressors. So the phage doesn't immediately take over to make the cell into a phage factory. The host can replicate, and daughter cells and dormant cells carry the phage genome around with them. This stable, heritable form is called a prophage. Upon phage induction, where that prophage cycle is stopped, it will switch into the lytic cycle of production. So it's this bursting or lysis of the host cells that is often used as the basis of culturing and isolating phage particles. So this picture plate here on the side is a double layer agar plate. I'll talk a bit more about that later. Essentially, there's a monolayer of bacterial hosts, and where a phage has replicated, there's a clearing zone called a plaque. I've included this photo as it's in no way publication quality, but it shows that there's a mixture of phages there, as you can tell by the different size plaques. And there's also a big fat air bubble there, so that's definitely not a phage, but you may see it in your plates. And these are other air bubbles because the organism creates gas and it's trapped underneath the plate. So that's just a couple of practical things to look out for. So the phage plaques can come in all different shapes and sizes. It's not a great diagnostic tool, but it is a way of working out what different types of phages you have there. There's also broth culture where a phage infects and can completely lyse the bacterial cells that are there, which is what you see in this diagram over here. So if you want to culture phages and get some cool results like these, what do you need? Well, most rumen microbes, as was probably said a few times already as part of this webinar series, require anaerobic conditions to grow. So you need a laboratory infrastructure and specialist capacity to do this. Your laboratory will need basic physical containment, sterile aseptic work areas. It needs to satisfy usual biological safety requirements. The equipment, media and skills to grow and store the microbial host. So effectively anaerobic culture capacity if you're working with rumen strains. Your experimental requirements, you'll need a microbial host and some phage stocks. If you don't have any phage stocks, you need to do isolations to get some. And for that, you'll need some phage source material, and if you're working with rumen samples, that will include all the relevant permissions to obtain them. This is an example laboratory equipment. This may have been covered yesterday, but so this is some stuff in our lab. So we've got an anaerobic chamber, which is a koi one, and gas piping to allow culture media to be made and everything. Our lab is set up for using Wheaton bottles and serum bottles, and the screw thread Hungate tubes. I understand that a lot of particularly American labs use the Bolsch tubes, which is shown here as well. We incubate. Our anaerobic chamber is run at room temperature. Sometimes people might have a little incubator inside their chamber, which is very good for the plate work. But we instead use anaerobic canisters. There's a range of commercially available plastic canisters, which we've had various degrees of success with. For really slow growing organisms, we do prefer the steel canisters. The original design was by Bolsch. That's the picture from the original 1979 paper, and I always think it looks impossibly tall. But anyway, ours is designed for this. Ours is above this with a plate beside it to give scale, and it's designed to fit in through the airlock of the anaerobic chamber. So this slide shows the basic steps of phage isolation, and it's undertaken in aerobic conditions. So the majority of phage work has been done in aerobic conditions because a lot of the organisms that have historically been of interest are aerobes, including, and phages are increasingly being investigated for their use in controlling nasty hospital infections and things. And most of those bacteria are aerobic in nature. So I borrowed this from a lovely publication, which I recommend people look at if they're considering doing this kind of work. So the first steps is with your sample, getting rid of any bacteria that might contaminate your experiment. Filter the sample to get rid of them. In this case, they're using wastewater. You should always use low protein binding filters. At the same time, you've got your host strain growing. There's always exceptions to these rules, but generally bacteria are more susceptible to phage infection when they're in the log phase culture, log phase part of their growth. And then you take your sample and you take your culture and you bring the two together into some melted soft agar, quickly pour that onto a base plate of more solid agar, incubate it, and hopefully you can then count the plaques. So that looks all very good with a nice little biorender generated figure. But in the anaerobic microbiology lab, it's probably going to have to be scaled down a bit. So here we have a source sample, which we've pulled from the freezer. If you're isolating from rumen fluid, you're probably going to have to centrifuge and clarify it before you can run it through the filters because everything will clog up. Then you have your host strain that's ready to go in its log phase state. You bring everything through the airlock, including your molten agar. So you may have to have it a little bit warmer so it doesn't set like a rock as it comes through the chamber airlock. And then you can do your plating in the chamber. So I've added some additional steps here. You can then pick the plaques. You might, if you're working with an anaerobe, you're going to have to do that in the chamber as well. You can then bring everything into the broth culture and make your stocks. So I've just put in a few additional steps here because the basic plaque assay can be optimized according to what your sample is. So you can have sample enrichment, and the theory behind enrichment, perhaps a little like what the fungi require, is that if you take your sample and you mix it with the host that you are interested in, then it will effectively multiply up the number of phages available because they go through a replication cycle and effectively amplify. You can concentrate your source samples. That might help concentrate the phage particles, particularly if you're using something like wastewater. You can include divalent ions because divalent ions are sometimes essential for phage attachment to cell walls. At the end of the day, it's really important that you understand the biology of your host. You need to really find the best growth phase for optimal infection. And just a few practical tips there listed out. For anaerobic hosts, you need to keep the clarified source material either very low volume or anaerobic because you have to mix it with your culture. Or plasticware, you have to allow time in the anaerobic chamber to reduce the oxygen contamination. If you're using canisters, this is important. Keep the agar warm for plating because the airlock cycles decrease the temperature. And always practice really good sterile technique when you're using bacteriophages or viruses because they can contaminate your lab, and then it will be very difficult for you to grow your bacteria again because they could pick up the particles and end up dying. So often for filtration steps that don't have to be anaerobic, they might be undertaken, say, in a biohazard hood cabinet, which you can then irradiate at the end of the day. And always include non-infected host-only controls to just double check if you've got any contamination coming into your lab. So a little bit about broth lysis. Broth lysis is also very important for making your stocks, and it can be scaled up, and it is the basis of making enough particles so you can do nucleic acid extraction if you're going to do any genome sequencing or protein analysis. So I've just listed a few of the steps here and the uses of the lysis broth culture. Very important to note is if you're going to do anything with phages afterwards, like any kind of animal trial or anything like that, you have to be aware that phage replication can accumulate a high amount of toxins in the lysates. So that's things like endotoxins, exotoxins, soluble bacterial proteins, and peptidoglycans. At the end of the day, the virus has blown that bacteria apart, and everything will still be there in the culture. So for in-animal applications, phage preparations must be as pure as possible because particularly the endotoxins, they're highly toxic and can lead to bloodstream infections and septic reactions. So for phage characterization, you require concentrated phage particle preps, free of host materials, so free of host proteins and nucleic acids. But once you've got that, there's a lot of things you can do. You can do electron microscopy to better understand the morphology, whole genome sequencing. At the end of the day, phage genomes aren't funky genomes. They're really pretty small. You can even use some even old tech with short reads, and you'll probably get the whole genome sequence out, no problem. The annotation of viral genes may take a bit of detective work. At the end of the day, as COVID showed us so nicely, viruses are very prone to mutation. DNA viruses are not as bad as RNA viruses, but you may have to do more protein homology to annotate your genes than nucleic acid homology. And once you've got your free phage particles, you can do protein chemistry, proteomics, etc. It's also good to know some of the biology of your isolates. With cultures, you can really better understand phage-host interactions, the attachment infection mechanisms, and determine the host range, like how many hosts will your phage infect? Will it infect everything in the rumen, or will it affect just one genus, or will it only infect one strain of one genus? They can be quite host specific at times. You can also examine host changes following infection. So CRISPR were first discovered with bacteriophages. So there's genetic mechanisms and physical mechanisms of resistance. Some of the physical changes you can very easily see by even over-incubating your phage culture with the host. You get, like you can see in this diagram here, you can get clumps forming of your bacteria. Effectively, they're trying to make a biofilm and stick together and exclude the phage particles. Once you've got cultures, you can also examine how the particles survive both in the rumen but in the greater environment. So that brings me to the knowledge gaps and really how long is a piece of string, but I've just jotted it down a few ideas here. So it'd be great if we could better encompass the phage or viral diversity in the rumen. So for this, we really need more isolates introduced into culture collections, including viruses infecting common rumen-associated prokaryote and eukaryote genera, and more complete genome sequences in the databases. The consequence of having more genome sequences in the databases is that you'll get better viral gene annotation, better assignment of taxonomy, because the taxonomy for viruses has now shifted to include both the particle morphotype, like whether it's a tail virus or not, and genetics. And currently, most of the rumen phage genomes that have been published don't fit well with those new classifications. Oh, excuse me, my lights turned out in the room. Anyway, we can also get better prediction of phage-host interactions and increase the accuracy of viral metagenomic binning. And we also need cultures to better understand rumen phage or virus biology. There are some genera of rumen microbes that are maybe more susceptible to viral infection to others, but we don't really know that. Parameters determining phage infection. Phage host prediction is so much easier if not only you have integration genes, but you have cell membrane attachment sites known as well. Burst size, the number of particles that are produced with each replication cycle, like some phages may be better at forming more particles than others. Phage life cycle, knowing how they survive, how they spread, how they establish, and any biofilm interactions. And I just put this last one in to be a bit of a stirrer. Do they really contribute to gene flow in the rumen? So quite likely that they do. But a lot of the information that's feeding into the answering this question at the moment is based on virome studies, and a culture will tell you so much more in this space, even if you're just using model ecosystems. So just to finish off, I'll thank our lovely team at in the microbial ecology group. So we're based at the Eco Sciences building at Dutton Park, and these are the permanent staff in our group. Our team is run by Diane Auerkirk. Thank you. I might just stand up for a moment and try and get my lights working again. Oh.
We will wait for Ross that she can turn on the lights because
Oh, I beg your pardon.
yes. And I asked the audience to type in your questions in the question and answer text block.
I'm so sorry about that.
That's perfect. At least we can see you again. They want that you stop working.
Yes, this is it. These buildings are so energy efficient.
Which is good. Good. So we have at the moment one question. Do you see it, or shall I read it for you?
So if I press this little Q&A, could phage bring disease to human beings? Ah, that's a big question. Well, there are a lot of examples where phages have brought disease to human beings. So particularly the lysogenic phages, there's some very famous examples of this. Botulism toxin is a phage-encoded gene. Cholera, that's formed by phage genes. The Shiga toxin genes are carried around of E. coli. They're carried around. At the end of the day, when you work with any microbe in the laboratory, you should be careful. So, yeah.
May I ask one question?
Certainly.
You were talking about anaerobic conditions, so yeah, it's mandatory for rumen microbes, but I wonder whether it was because of the host or the phages, they are sensible to oxygen.
It's mainly the host, I think, actually, and because it's a little bit chicken egg, you can't grow the phage without the host. You should always provide the ideal conditions for growing the host. I guess by shifting into aerobic conditions, some facultative or aerotolerant bacteria can still be growing, like the Streptococcus bovis or Equinus is a very good example of that. It can tolerate oxygen, but whether growing it in conditions where there's oxygen changes the host, it probably would, because it's using different metabolism. Whether that would change the susceptibility to phage infection or not, well, that's an unknown, but it could occur.
I have also a question in respect to the success of the isolation. Do you know if there is a certain moment of the lifespan of a cow that you should take the rumen sample because there are a lot of phages inside, or do you see it because we know that there is a development on the rumen microbiome from the calves to the cows and during the lactation and non-lactation period? So is there any conclusion already known that maybe a lactating cow is a better donor to provide bacteriophages, or is there any systematic studies already done?
No, I don't know of that. At the end of the day, the viral populations are really heavily linked to the host populations. So where you would see, where you have perturbation like changes to the microbiome, you will also see changes in the viral population, and that's been shown across a lot of ecosystems, like in seawater and things like that as well. I think it depends on the concentration of your host that is there. So in the newborn calf that is on a milk diet, there'll be a lot more lactobacillus, say. So that would be a good place to look for lactobacillus viruses, because there's a lot of their host there. That may shift as the rumen evolves and the animal is on grass or whatever, and the rumen evolves. So it really just depends on what kind of virus you're looking for. Does that answer your question?
Yes, thanks.
Thank you.
And we have another question in the QA from Johan.
The question is, how reliable do you think are the phages that we are currently assembling from viromic studies? Well, I guess all assembly has its limitation. I think if there is, it's a coverage question often in bioinformatics, isn't it? If you've got good coverage, then you can it increases the confidence in the genome sequence information that you're assembling. I think probably the problem with some of the viromic studies is the annotation of the contigs, viral contigs that you're assembling, how complete they are, perhaps. You won't. That's a hard question to answer at this moment in time, because there isn't a lot of rumen phages in the databases. Does that help, Johan? How would you look at phage if you're chosen? Oh, sorry, that question disappeared on me. How would you look at phage if your chosen host wasn't forming a nice opaque layer in plaques? Well, that's getting a bit tricky, isn't it? Well, you could shift to lysis cultures, I guess, but having even a kind of layer on your plates would help, very much help for finding a phage particle. If your host clumps and doesn't grow well, perhaps look for a strain that doesn't clump and try that one.
Okay. Is there any other question? Can wait for some seconds. There's one.
So the next question is, phage therapy has been suggested for various infectious pathogens, and even in some cases as methane mitigation approach. What is your view on organisms developing CRISPR-based immunity against phages in the long term? So I think there's almost two parts to this question. Phage therapy has indeed been suggested for infectious pathogens, and there's commercially available formulations for reducing pathogens based on phage therapy. There's also customised phage therapies developed, say, for individuals who are chronically ill with bacterial infections that are impossible to shift with the current antibiotics. So that personalised medicine is a thing. It's usually a last resort. But yes, indeed, that is occurring. The second part to the question, they have been suggested as methane mitigation approach. There's a couple of ways you can control the methanogen population with phages. The first one would be with whole phage particles, similarly to the medical application. The other way is to use the enzymes that they encode to help break them apart, that's called enzybiotics, where the enzymes are used as an antimicrobial. So what's my view about CRISPR-based immunity against the phages in the long term? It may well occur. At the end of the day, all phage therapies tend to use a cocktail of phages. They don't just stick to one. They tend to throw in quite a few to help overcome resistance effects. I think at the end of the day, you have to be careful drawing parallels between what can occur quickly in the laboratory and what can occur inside a cow. So in the cow, you've got outflow and all those biological parameters in the rumen. In the laboratory, you can get CRISPR developing very quickly. In fact, that's how people found them in the first place. I hope that answers your question. How successful are phage-based methane mitigation strategies? Well, there hasn't been many developed as such, so it's a bit hard to say how successful something is that hasn't been developed. I hope they are successful.
So thank you very much, Ross. I think there are no more questions. I don't know, Milka, you want to wrap up the webinar? Or?
Well, I just want to say thank you to all the speakers, all the people that we had online. It was a big pleasure to see that everybody was interested. So thank you very much. And yeah, I think it was very nice presentations. And see you next time.
Thank you very much. Have a nice day or good night.